Good morning, everybody. My name is Stefan Pӧhlmann. I'm working at the German Primate Center. It's my pleasure to talk to you about SARS coronavirus 2 entry into cells and its inhibition. The idea is to briefly introduce the coronavirus zoonosis and the coronavirus spike protein, and then talk about the proteolytic activation of the viral spike protein and its inhibition. And finally, in the last part of the presentation, I will introduce the variants of concern and talk about their impact, or talk about the impact of mutations in their spike proteins on inhibitor and antibody sensitivity. I think I can be very brief on this slide. Coronaviruses, as you all know, are enveloped RNA viruses. They have three surface proteins, the M protein, the E protein, and the spike protein. And the spike protein is actually large enough to be seen by electron microscopy, as indicated on the left side. They are providing the particles with this crown, this corona-like appearance, which coined the name for this family of viruses. It's not entirely unfair to say that interest in coronaviruses a couple of decades ago has been moderate. And this is why. It has been well known that coronaviruses infect animals and cause severe diseases that are associated with significant economic losses. However, it was also clear that they are human coronaviruses, but they don't do a lot. They circulate worldwide. They can even cause reinfections, but it was known that they only cause the common cold. And what has been learned in a very hard way during the last decades is that the coronaviruses from animals can be zoonotically transmitted to humans, and that this can have severe consequences. And this was realized the first time during the SARS pandemic in 2002 and 2003, when the virus was transmitted from a bat reservoir via an intermediate host, probably civet cats, to humans, and infected humans developed a new respiratory disease called severe acute respiratory syndrome. This respiratory disease was associated with a case fatality rate of 10%, which was, however, strictly age-dependent, with older individuals being mainly afflicted. Another characteristic of SARS, or SARS-1, was that hospital personnel was frequently affected, and the vast majority of cases occurred in Asia. And key point, fortunately, the transmissibility of the virus between humans was moderate, and therefore the pandemic could be ended in the absence of specific vaccines and antivirals. And the next coronavirus outbreak, in fact, did not occur in Asia, but in the Middle East, and was caused by the MERS coronavirus that infects bats and has been transmitted from bats to camels a long time ago. Now camels are reservoir hosts and transmit their viruses to humans. And infected humans develop the Middle East respiratory syndrome, MERS. It's again a respiratory disease associated with a high case fatality rate. And again, as for SARS-1, hospital personnel is frequently affected. Transmissibility between humans so far is fortunately low, but there are constantly new cases, so the virus is still around, and there's concern that it might evolve towards high human-to-human transmissibility. Let me briefly summarize what I said. This is indicated here on the left side. We do have human coronaviruses that are ultimately of animal origin, but now they exclusively circulate in the human population. They do that worldwide, and infection is associated with the common cold. And what we have seen in the last decades is the transmission of animal coronaviruses to humans. Specifically, SARS-CoV-1 and MERS-CoV have been transmitted via animal hosts to humans, but transmissibility between humans was moderate to medium, and this is why we didn't see large-scale pandemics. And very unfortunately, This has changed in 2019, 2020, when, as you all know, another animal coronavirus, SARS coronavirus 2, was transmitted from an incompletely understood animal host to humans to give rise to the new lung disease COVID-19, and subsequently that virus spread pandemically to cause the COVID-19 pandemic that has dramatic social, economic, and medical consequences, and for which countermeasures, specifically the development of antivirals, is urgently needed. And my lab, in order to do so, is focusing on the coronavirus spike protein. So next I will briefly introduce you to coronavirus spike proteins. We already said spike proteins sit or are inserted in the viral envelope, and they have one major task, and that is to recognize target cells and to allow the virus to bind and enter those target cells. And for this, the spike protein interacts with cellular receptors, and in case of SARS coronavirus 1 and SARS coronavirus 2, this cellular receptor is the ACE2 protein. However, receptor binding is insufficient for entry. In addition to receptor binding, the spike protein also needs to be activated by a host cell protease, and that protease is the cellular enzyme tempress 2. So in brief, you can imagine the spike protein as the access key of the virus to the host cell. The receptor is kind of the entry door, and the protease is the activator of the entry key. Now, on a more molecular level, this is how coronavirus spike proteins look like. They are trimers. They are subdivided into units, a surface unit S1 and a transmembrane unit S2. And those two units have very different tasks. The surface subunit S1 has the task of binding to cellular receptors like the ACE2 domain, like the ACE2 protein. And for this, the S1 subunit harbors a receptor binding domain, and inside that receptor binding domain, a receptor binding motif that makes direct contact to the receptor. In contrast, the transmembrane unit S2 has a different task. It is responsible for fusing the viral membrane with the cellular membrane that allows delivery of the viral genetic information into the host cell cytoplasm. And that is, of course, essential for infectious viral entry. However, in order to do so, the spike protein has to be cleaved by cellular proteases, and two cleavage sites have been identified in the spike protein. They are called S1, S2, and S2 prime. And since cleavage is essential for infectious entry and is carried out by cellular enzymes, those proteases, are potential targets for antiviral intervention. Now, when at the beginning of the pandemic, the spike sequence, protein sequence became available, it harbored two major surprises. It was pretty obvious that the receptor binding domain of SARS-2 spike was quite similar to that of SARS-1 spike, immediately raising the question whether it would bind to ACE2, which subsequently we and others showed that it does. The second surprise was the sequence of the S1, S2 cleavage site. That cleavage site harbors several arginine residues, which are a signature of high cleavability, and such a optimized cleavage site is not found in very closely related coronaviruses. And that even stimulated and still stimulates discussions, suspicions, whether the virus arose from a laboratory. In the meantime, we've learned that several animals, specifically pangolins and bats, harbor viruses that are closely related to SARS coronavirus 2. And not only that, it is clear that the unexpected ACE2 binding site is found in some of those viruses, and some even have alterations at the S1, S2 site that might increase cleavability. And that suggests, also it does not definitely prove, that SARS coronavirus 2 is of zoonotic origin. And in fact, it is worthwhile stating that bats harbor SARS-CoV-2 related viruses that are antigenically sufficiently different from SARS coronavirus 2 to probably not be efficiently inhibited by antibodies raised against SARS coronavirus 2 infection. Therefore, from my perspective, it is not so much a question whether a SARS coronavirus epidemic or pandemic repeats itself. It's more a question when this will happen. Now, in order to come up with counteractive measures that allow for protection against infection with SARS coronavirus 2 and also related coronaviruses, we are focusing on the proteolytic activation of the viral spike protein. And we are trying to inhibit this process. And in the next couple of slides, I'll introduce the progress that we've made in this field. And before doing so, it is my pleasure to recognize or to state that most of the data that I'll be presenting have been generated by a very skilled postdoc in the laboratory, Dr. Markus Hoffmann. And what Markus and colleagues in the laboratory did first is to establish a model system that allows analysis of SARS coronavirus 2 entry into host cells without particular biosafety measures. The system is based on a vesicular stomatitis, short VSV, based vector system that can acquire the spike protein of SARS coronavirus 2, and when doing so, can transduce cells a single time, and transduction efficiency, meaning entry efficiency mediated by the SARS coronavirus 2 spike protein, can be easily quantified. And now the system is used in many laboratories to study entry and its inhibition. Now, Markus did put that system to use to analyze how the spike protein is activated by host cell proteases. And our major findings are actually summarized in this slide. We found that the spike protein is cleaved first in infected cells, specifically during the passage through the constitutive secretory pathway. Spike is cleaved by the host cell protease furin, and cleavage occurs at the S1, S2 site. This cleavage is required for viral spread in the lung, but it's not the only cleavage that is required. A second cleavage that is essential for entry into lung cells is carried out by the cellular protease TMPRSS2. It's again believed to be important for viral spread in the host and occurs at the S2 prime site. In addition, there's an auxiliary activation route in many cell cultures. The virus can be taken up via endocytosis and is then activated in the late endolysosome by the host cell proteases cathepsin B and L. However, we believe that this is an activation route available in cell culture, but not in the infected host. And those findings have consequences regarding therapeutic options for treatment of COVID-19. You remember chloroquine and hydroxychloroquine have been suggested, actually have even been advertised for treatment of COVID-19. And we feel that this is not a good idea because chloroquine and hydroxychloroquine block this auxiliary activation pathway that is dependent on cathepsin B and L, and that we believe is only operative in irrelevant target cells. This is maybe best analyzed by the data shown to the right. when we look at virucells, which are very frequently used in the laboratory to propagate SARS coronavirus 2, they do not have TMPRSS2, but they do express cathepsin L. And here we see very efficient inhibition by chloroquine and hydroxychloroquine. However, if we do the same inhibition experiment with KLO3 cells, which is a lung-derived cell line that has endogenous TMPRSS2 expression, we see very poor, if any, inhibition by chloroquine and hydroxychloroquine, strongly indicating that those two substances will not be suitable for COVID-19 treatment. And that conclusion is actually supported not only by animal data, actually different animal models have been analyzed, but also by data from human clinical trials. However, our research did not focus on the auxiliary activation pathway. We focused on the TMPRSS2 dependent pathway. And we were excited about this because an inhibitor was available, camostat mesylate, that was previously found by other groups to inhibit TMPRSS2 dependent entry and to block TMPRSS2 activity. And indeed, when we compared inhibition of MERS, SARS, and SARS-2 spike-driven entry, we saw that all of those entry processes were sensitive to camostat, whereas entry driven by a control protein, the G protein of VSV, was not blocked. In addition, we found that camostat efficiently blocked replication of authentic SARS coronavirus 2 in lung cells. And that is exciting, or was exciting to us, because camostat is used for decades in Japan for treatment of pancreatitis, suggesting that it might be suitable for COVID-19 treatment, suggesting that repurposing is an option. And because of that option, we continued to study camostat and TMPRSS2, and in the recent months, we addressed the following question. We asked whether TMPRSS2-related proteases can also activate SARS-2S and whether those proteases are blocked by camostat. In addition, we asked whether camostat blocks SARS coronavirus 2 spread in the human respiratory epithelium. And finally, of course, we are interested to know whether camostat is active in animal models and humans. And in the next slide, I'll try to answer those questions. First of all, are there TMPRSS2 related proteases that can also activate SARS coronavirus 2 spike? In order to address this, we used one of those cell lines in which entry depends on the auxiliary cathepsin L dependent pathway, and we blocked this pathway by ammonium chloride. And in parallel, we exogenously expressed the proteases indicated here, and then we asked whether they would be able to rescue virus from the ammonium chloride installed block. TMPRSS2 did that very efficiently, as expected, but it was not the only protease that did so. Also, TMPRSS11D, E, and F, as well as TMPRSS13, allowed for activation of the viral spike protein. Now, that was exciting to us, and in order to get first insights on whether those, let's call them auxiliary proteases, might play a role in SARS coronavirus 2 spread in the infected patient, we asked whether they are expressed at the right place. And for this, Joan Smith and Jason Schelzer in New York City used single-cell RNA-seq analysis of the human lung. And here are their major findings. When looking at the lower respiratory tract, ACE2, this is the upper panel, and TMPRSS2 expression was readily detected, and substantial overlap in expression, meaning co-expression, was found. However, the other proteases were not efficiently expressed in the lower respiratory tract, with TMPRSS13 being a somewhat an exception, with some TMPRSS13 expression being detectable. So, based on those expression data, it is fair to assume that spread in the lower respiratory tract might be mainly TMPRSS2 dependent. Interestingly, for the upper respiratory tract, the situation looked different. Again, good co-expression of ACE2 and TMPRSS2 in relevant target cells, but in the upper respiratory tract, also good levels of TMPRSS 11D and 13 were seen, suggesting that the virus might have the choice from several activating proteases when spreading in the upper respiratory tract. And of course, this also raised the question whether those additional activators of the spike protein would still be blocked by camostat. And the brief answer is yes. They were all efficiently blocked by camostat in the previously described rescue assay. Now that looked good so far. And the next question was, well, all the previously shown results were obtained with cell lines. How about primary human lung cells? And we addressed this question jointly with Armin Braun at the Fraunhofer ITEM in Hanover, who kindly provided precision-cut lung slices, meaning slices of human lung. Those slices were cultured and infected with authentic SARS coronavirus 2 in the presence of camostat, a metabolite of camostat that is commercially available under the brand name 4251. And we also included a related inhibitor, also a protease inhibitor that targets TMPRSS2, and that is called nafamostat. And particularly when looking at the 5 micromolar concentration, we found that camostat and to somewhat higher efficiency nafamostat very appreciably blocked viral spread in this tissue. And those results have also been confirmed by others in closely related systems. So this made us quite excited about the options of camostat for COVID-19 treatment, but unfortunately, recent months brought significant disappointment, and here's the current situation. In mice, there's now, from my perspective, strong data that TMPRSS2 is, as expected, required for efficient spread and pathogenesis, and there's also data from the mouse model indicating that camostat, which is the black line here, and to a higher degree, another TMPRSS2 inhibitor called aboralstat can protect mice from weight loss upon SARS coronavirus 2 infection. So that looked all promising. However, recently results from a well-controlled clinical trial in Denmark were reported, and here no statistically significant difference between placebo and camostat was observed, which is of course disappointing and raises the question whether TMPRSS2 is the right target. When looking at the left side and additional papers that are currently coming out, I still feel that TMPRSS2 is absolutely the right target. However, the question whether inhibitor has been given too late and whether sufficient amounts of inhibitor became available in the human lung are still open, and they urgently need to be answered. And before ending the TMPRSS2 part of my presentation, I would like to point out that other clinically relevant inhibitors of TMPRSS2 have been discovered, and noteworthy might be alpha-1 antitrypsin, which has been shown to inhibit TMPRSS2 activity. This is indicated by the findings presented in the left side. And alpha-1 antitrypsin has been shown to reduce SARS coronavirus 2 infection in cell culture. And first initial evidence has been provided that alpha-1 antitrypsin might be protective in the context of SARS coronavirus 2 infected humans. So TMPRSS2 research exploiting this protease for COVID-19 treatment is still ongoing, and some successes, at least on this level, have been reported. Let me summarize what I just said. So I told you that TMPRSS2 related enzymes activate the SARS coronavirus 2 spike in cell culture and may contribute to viral spread in the upper respiratory tract. Those enzymes are all blocked by camostat, and camostat also blocks SARS coronavirus 2 infection of human respiratory epithelium ex vivo. There's more and more evidence that TMPRSS2 supports viral spread in mice, and there's even data indicating that camostat protects from weight loss upon SARS coronavirus 2 infection in the mouse model. However, in humans, camostat treatment did not provide COVID-19 patients with a significant benefit. And finally, there's data indicating that alpha-1 antitrypsin blocks TMPRSS2 and also blocks SARS coronavirus 2 infection. And now I would like to switch gears and move away from TMPRSS2 and move towards the variants of concern. I think they don't need a lot of introduction. The virus has been genetically relatively stable over some time. Just the D614G mutation became dominant early on. But more recently, variants keep emerging and are subdivided into variants that are of interest and variants that are of concern. And drivers of this development are, of course, augmented transmissibility and immune evasion. And when this development started, we focused our research on the variants B117, the UK variant, B1351, the South Africa variant, and the P1 variant from Brazil. All those variants harbor mutations in the spike protein. Some of them are located in the receptor binding domain and might change interactions with ACE2 and, of course, recognition by antibodies. And therefore, we looked whether those spike proteins could still be efficiently inhibited by therapeutics and antibodies. Here are our results. Regarding potential therapeutics, we looked at soluble ACE2, at TMPRSS2 inhibitors, and inhibitors of membrane fusion. And the good news is all of them work fine against the variants of concern. There's really no appreciable difference regarding soluble ACE2, camostat, nafamostat, and membrane fusion inhibitors. Actually, one variant is even better inhibited than the wild type. So this is good news. But news were not as good when looking at antibodies. We looked at several. One of them is bamlanivimab, which received emergency use authorization for COVID-19 treatment and has also been ordered in Germany for that purpose. However, if we look at B1351 and P1, we see that those two spike proteins are resistant to bamlanivimab, indicating, and I think this is now common practice where possible, that it is preferable to use two antibodies simultaneously for treatment, because if a single antibody is used, there's the considerable risk of resistance development. So in short, the B1351 and the P1 variants are bamlanivimab resistant. Now, how about resistance towards antibodies generated by infection and by vaccination? We also looked at this question using sera from COVID-19 patients that were kindly provided by the local doctors at the University Hospital Gøttingen, particularly Martin Sebastian Minkler, and they were used in our neutralization assays, and this is what we found. Regarding B117, the UK variant, we found that this variant is highly susceptible to antibodies, and we feel, and I believe everybody agrees, that the rapid spread of B117 is mainly driven by increased transmissibility and not so much by evasion of antibody responses. However, if we look at B1351 and P1, situation is quite different. Particularly for B1351, we see a marked reduction in neutralization sensitivity that is statistically significant. And we see a similar effect for P1. Also, the effect is not as prominent as for B1351. Sorry, the situation for vaccination is not very different. We also tested sera from patients that had received 2 shots of the BioNTech mRNA vaccine. And again, we looked at neutralization and found similar phenotypes. B117 was again nicely inhibited by antibodies induced by that mRNA vaccine. In contrast, inhibition of P1 and particularly B1351 by antibodies was clearly reduced, indicating that B1351 and P1 are really immune evasion variants. And what I just said is summarized here. So antibody-mediated protection after convalescence and vaccination is high for B117, but is reduced for B1351 and P1. And the same applies to the effectiveness of therapeutic antibodies like bamlanivimab. Now, to the end of this presentation, I would like to focus on the variant that is currently spreading in many countries and is raising a lot of concerns. That is the India variant, the B16172 variant. This variant became recently dominant in India. Look at the prevalence in the recent months, which is skyrocketing. And as you all know, this variant became also dominant in the United Kingdom and is already dominant in Germany as well, and probably also in other European countries. And of course, this again raised the question whether there's anything particular about that variant. And we first addressed this question by asking whether the spike protein of the India variant is particularly suitable to fuse lung cells. And just as a background here, if infected cells express the spike protein, well, infected cells do express the spike proteins, and this allows them to fuse with neighboring cells, resulting in the formation of giant cells that are called syncytia. And there's evidence that syncytia formation occurs in COVID-19 patients and may contribute to COVID-19 pathogenesis. And when we looked at syncytia formation for B16172 spike, we found that syncytia formation was markedly increased. And when counting syncytia, this becomes obvious here. It's about the B16172 spike. It's about 2-fold more efficient in generating syncytia as compared to the wild-type virus. And we believe that this is most likely linked to this mutation at the S1, S2 site, which likely increases cleavability by furin, and this likely translates in the increased cell-to-cell fusion. And this question is currently investigated in the laboratory. Now, how about entry into cell lines, and how about ACE2 binding? When looking at the standard cell line Vero that I previously introduced, we don't see any differences in entry efficiency, and the same applies to the also frequently used cell line 293T. Entry into both cells depends on cathepsin L, and again, there's no difference. However, if we look at two cell lines that allow for TMPRSS2 dependent entry, the colon-derived cell line KCO2 and the lung-derived cell line KDO3, we see that the B16172 spike clearly mediates more efficient entry into those cell lines. And importantly, this more efficient entry might not be due to increased ACE2 binding, because if we measure binding of soluble ACE2 to spike protein expressing cells, we don't see a major difference. And at present, we speculate that the increased lung cell entry might be due to usage of attachment promoting factors that are specifically expressed in lung cells. And which factors those might be is an area of active investigation. Finally, how about antibodies? Again, bamlanivimab and related antibodies were studied, and the simple answer is the virus was sensitive to inhibition by 3 out of 4 antibodies, but it was insensitive to inhibition by the previously mentioned bamlanivimab. So this antibody is not suitable for treatment of B16172 infected patients, at least as monotherapy. How about antibodies induced by infection and vaccination? We analyzed this using the B1351 spike as a control, and we can see some level of immune evasion, both for convalescent sera and from sera from vaccinated individuals. But it's fair to state that immune evasion is not as prominent as that observed for B1351. And before closing up the variants of concern section, I would also like to point your attention to the fact that variants of concern, or SARS-CoV-2 variants that harbor mutations that allow for immune evasion, might not only emerge in humans, but can also emerge in animals. Thus, you might remember that SARS coronavirus 2 can be transmitted from humans to farmed mink. And in the infected animals, the virus can acquire mutations, and from the infected animals, it can be transmitted back to humans. And we asked whether mutations like the one shown here that arise in infected mink modulate neutralization by antibodies, and our findings briefly indicate that yes, this is the case. Viruses that emerge in mink can be partially resistant to antibody-mediated neutralization. So it should be underlined that also infection of animals can result in variants that might not be fully controlled by the human immune system. Let me summarize this part of the lecture. I showed you that B16172 variant enters and fuses lung cells with increased efficiency. It's resistant against the therapeutic monoclonal antibody bamlanivimab, and it's partially resistant to neutralization by sera from COVID-19 patients and sera from mRNA vaccinated individuals. And finally, SARS coronavirus 2 can infect mink, and in those animals, the virus can acquire mutations that reduce antibody-mediated neutralization. With this, I would like to thank the people in the laboratory, particularly Markus Hoffmann, who did the work that I presented, but also many thanks to Hanna Kleine-Weber, Heike Hoffmann-Winkler, Nadine Krüger, Prerna Aurora, and Inga Nehlmeier and colleagues. And of course, many thanks to our colleagues at other institutions like Frank Nøe in Berlin, Mats Kjølby in Aarhus, Denmark, Jason Schelzer at the Cold Spring Harbor Laboratories in New York, Olga Danhoff and Armin Braun, the Fraunhofer in Hanover, Germany. Of course, Marcel Müller and Christian Drosten at the Charité in Berlin. Oops. Not sure what happened now. Here we are. Martin Sebastian Winkler in Göttingen, Jan Münch and his crew at the University Hospital in Ulm, and finally the Deutsche Forschungsgemeinschaft, Bundesministerium für Bildung und Forschung in the country of Lower Saxony for financial support. And thank you very much for your attention.
Stefan, thank you very much for a very interesting talk. We have time for some questions, and I will take the opportunity that I'm a German and I will ask the first question. Stefan, you showed a lot of information about the genetic diversity of SARS-CoV-2 and the neutralizing activity of the antibody. But do you think that we really should put so much attention of the new genetic variants of SARS-CoV-2 if we have the confirmation that the vaccine is still working? Even though that the neutralizing activity is lower, the vaccine is working and it's quite efficient. And if you compare it, and can you compare it to the genetic diversity of the common cold coronaviruses or the genetic diversity of the MERS coronaviruses? Do you know anything about the variants of MERS-CoV-2? If this emergence of the genetic variants of SARS-CoV is something completely new for the coronavirus, or is it something what we observed in the previous epidemics?
Well, thank you for the question. Regarding vaccines, yes, absolutely, and I didn't mean to misguide you. So, of course, the message should be that the mRNA vaccines are highly protective even against the India variant. However, I would not be surprised if the number of vaccine breakthrough infections, even for mRNA vaccinated individuals, would be slightly higher for the B.1.617.2 variant. However, this efficient protection may not be provided by other vaccines, including the vaccines based on whole-killed virus. So the situation in countries that use the mRNA-based vaccines might be different from that use other vaccines, particularly vaccines relying on whole-killed virus. So message from my perspective would be, yes, please rely on vaccines, but vaccinate fast. So the only way to keep B.1.617.2 in check is, from my perspective, fast vaccination, preferentially with an mRNA-based vaccine. Regarding further development, I can't say. I'm not sure whether we are at the end of resistance development, meaning if the viruses that are currently circulating are as neutralization resistant as SARS-CoV-2 can get. The fact that they harbor related and sometimes almost identical mutations would argue in this direction, but only future can tell whether viruses might emerge that show even increased resistance development. And you might have seen a recent paper, I think it now appeared, indicating that at least in some cell lines, SARS-CoV-2 can enter in an ACE2 independent fashion. And of course, this raises the question whether this is just an in vitro phenomenon or whether this may also occur in patients, and whether this impacts susceptibility to antibody-mediated neutralization. To be entirely honest, I'm not comfortable to make a strong statement regarding variants arising in the context of MERS or the common cold coronaviruses, but at least I'm not aware of such variants that harbor or that differ dramatically from, in brackets, wild-type virus regarding transmissibility, and antibody-mediated neutralization. But I could envision a similar process, but I'm not aware of a lot of data there.
Okay, thank you. And we have some questions from the chat from Jacek Kolonowski. Stefan, would you expect the antibody evasions like you showed in case of a protein-based vaccine, not the mRNA vaccines?
Yes, gut feeling would be yes. Also, immune evasion might occur. Whether when the antibody levels induced by a protein vaccine are significantly higher, and to be honest, I don't know if they are, then the situation might be different. But I would expect that the type of antibodies induced might be comparable, and then I would not be surprised if there's some level of immune evasion.
Okay.
But I'm on thin ice here because I'm not aware of many data regarding the protein-based vaccines.
And do you have a question from the audience? Julita?
Thank you for the excellent presentation. I would like to ask how the augmented entry of the SARS-CoV-2 variants to the colon cells may affect the human-to-human transmission and the course of infection.
Thank you. From my perspective, there's no evidence that SARS coronavirus 2 is transmitted via other routes than the respiratory route. So I would not be expecting that in more efficient entry into colon cells impacts transmissibility, but it might impact pathogenesis. So it is my information that ACE2 as well as TMPRSS2 is endogenously expressed in the gut and may support viral replication in this target organ. And if entry into gut cells is increased due to the improved, in brackets, S1, S2 side, I would expect, or I would not be surprised if the B16172 variant replicates more efficiently in this tissue, and that this might contribute to pathogenesis.
Thank you very much.
Stefan, and for the time's sake, we need to ask the last question, and it will be the question from the chat. As endocytosis pathway happens when TMPRSS2 activity is not there, would you expect to be a major path of infection if we inhibit activity of TMPRSS2 by drugs?
Yeah, so of course, I've been quite enthusiastic about the potential of TMPRSS2 inhibitors for COVID-19 treatment, and I've been quite disappointed by seeing our results with Camostat. However, when looking at preprints and mainly when looking at preprints, there's papers indicating that if TMPRSS2 inhibitors are topically applied, meaning are directly applied to the respiratory tract and are applied early in infection, they do very well in preventing virus spread and pathogenesis. So I'm still optimistic that TMPRSS2 inhibition is a good way to target the virus, but we might need to deliver the inhibitors directly to the respiratory tract, and we should treat as early as possible. And the latter statement, of course, also applies to most other antiviral strategies. And finally, if there's still time, from my perspective, it's important to point out that for a long time it has not been entirely clear why the virus chose to focus on TMPRSS2, considering that cathepsin L is broadly expressed, not only in cell lines, but also in the human body. But recent papers provide a nice explanation. They show that TMPRSS2 allows the virus to avoid an antiviral restriction factor that is present in endolysosomes. Those are the Ephetum-2-3 proteins. And a study even shows that Ephetum-2 is essential for interferon-stimulated suppression of SARS-CoV-2 infection. And if TMPRSS2 is around, the virus can avoid this block. So there's a strong rationale, a strong reason for the virus to rely on TMPRSS2 for host cell entry. Thank you.